Okay, so now we are finally going to get to this question. How does Ebola kill you? Okay, so with the 2014 epidemic, there was a lot of effort put into understanding the pathophysiology of Ebola, and specifically how it kills. And unfortunately, as we go through the pathophysiology, you'll notice that there are some similarities between how Ebola behaves in the body and how coronavirus SARS-2 behaves in the body, both with lethal outcomes. So the first question to answer is, well, what cells are picking up Ebola at the start of the infection? And it turns out that the first cells that Ebola infects are going to be mobile antigen-presenting cells. So in class we covered macrophages, but there's another group of antigen-presenting cells that are also very mobile, and these are dendritic cells. And dendritic cells and macrophages look very similar. You can see in the bottom picture that you have a dendritic cell, which is the blue one, activating a helper T cell in yellow. So if you remember your immunology, then these immune cells move around. And so this is a great way for Ebola to disseminate throughout the body systemically, because these macrophages and dendritic cells become infected at the portal of entry and then start moving around. Okay, so they get into the blood or they move into the lymphatic system, and then they start disseminating the Ebola virus. Now, Ebola picks a couple early targets. So in addition to antigen-presenting cells, Ebola also infects hepatocytes in the liver. It infects the spleen, and it infects the adrenal cortex. So the liver is going to be a major site of production of plasma proteins, including the plasma proteins that are involved in the coagulation cascade or the clotting response. In other words, they produce plasma proteins that help you make clots to prevent bleeding out of your blood vessels. The adrenal cortex is going to be a site where you produce a lot of hormones, including hormones that regulate the salt levels in your blood. That's aldosterone, and then also cortisol, which is the stress hormone, which is useful when you are stressed. So adrenal cortex cells being infected and therefore not doing their job is also not a good thing. Okay, you might have read on there that one of the big clinical presentations of Ebola virus is that it discombobulates our immune system by creating something called a cytokine storm. And this is similar to what COVID-19 does. So in COVID-19, critically ill patients often are experiencing what is known as a cytokine storm. Now, you have learned that cytokines are permission molecules. Now, I want to give you an analogy. Imagine that you have an army, and you have an army of 1,000 people. And all of these people, we'll do a prehistoric army, have 20 spears in their possession. And the goal of this army is to storm a castle or something like that. So we're going to storm a castle. We have 1,000 soldiers, all with 20 spears. And when they get up close to the castle, they all throw their spears at the castle. That would be analogous to a directed attack by our immune system, of which cytokines are like the spears. So we have, a certain amount of cytokines, they're useful, but they're meant to be directed at a real problem. When you get a cytokine storm, it's equivalent to taking those 1,000 soldiers and giving them 100 spears, not 20, but 100 spears each, to which you say, awesome, more power, right? But then telling them to just start throwing those spears randomly. Some of them might be thrown at the castle. Many of them will be thrown at each other, and you will get a lot of death, friendly fire, death, and you won't do what your objective is, which is to storm the castle, because so many of the spears are just being thrown around randomly that you don't have a coordinated response. And that is kind of what happens in a cytokine storm. You increase the level of your response, but it's totally uncoordinated and ends up causing more damage than good. And you usually are not able to kind of corral the forces to get a directed outcome, which in this case would be fight Ebola, stop running around fighting whatever. Stop throwing your spears randomly, remember the castle. So cytokine storms are very effective in discombobulating our immune system, and they usually have poor outcomes for us. Now, I've covered the early target cells. Those are going to be the macrophages and antigen-presenting cells. We know that those antigen-presenting cells are going to disseminate throughout the body, thus spreading the virus. And some other targets are going to be the liver, the spleen, and the adrenal cortex. And we know that one of the major features of this virus is that it induces cytokine storms, which effectively dysregulates our immune system's response to Ebola. So I want to talk about some other creative things that Ebola does. So if you were a virus and you wanted to replicate as much as you could in the human body, what are some of the things you would do? Well, I know what I would do if I was a virus and all I wanted to do was replicate. I would want to make sure that any lymphocytes that were trying to destroy me would be destroyed so that they couldn't mount an acquired immune response. If I infected a cell and that cell tried to warn another cell that I might be coming, I would want to shut that line of communication off. I would want any antibodies that would come out trying to find and coat me to go somewhere else or be rendered ineffective. And then, as I've been so successful, I need to get the heck out of this human before the human dies, or I won't be able to sustain my replication. So those are some things I would want to do, and Ebola does all of them. So Ebola is really, really good at replicating extensively in the body by suppressing our immune system, preventing cells from talking to each other, sending our immune system on a wild goose chase, and then it's also really good at getting out of the human body before that human succumbs. Okay, let's take a look at these one by one. So our first goal is to suppress the immune system, specifically the acquired immune system, which would be very helpful in helping the virus replicate unchecked if there's no acquired immune system to find infected cells or destroy the virus outside of the cell. So how do you inhibit the acquired immune system? It turns out that Ebola does not actually inhibit the acquired immune system as much as it delays it. So it delays it by reducing the counts of your lymphocytes, specifically your B and T cells. Now, your B and T cells are not infected, but they are still destroyed by something called bystander apoptosis. And bystander apoptosis is where a cell that is not triggered to be destroyed, just standing on the side, gets signals that, hey, you need to be destroyed. And so this is what happens. It's actually part of the cytokine storm, where infected macrophages and dendritic cells oversecrete certain molecules, including something called TNF-alpha. TNF stands for tumor necrosis factor. So that gives you a clue about what it's doing, necrosis, death, right? So this is a molecule that basically triggers the death of the nearby cells. And so it induces apoptosis. And so what that does is that brings the B and T cell counts really low early in the infection so that you don't mount an acquired immune response right away. And of course, that's going to help the virus replicate over and over and over again without fear of being caught. In addition, it turns out that our cells have kind of a warning communication system with a molecule that is known as interferon. And the name is exactly what it suggests, to interfere with something. So typically what happens is when a virus gets into our cell and starts replicating, we've already learned that one of the things that happens is we would express the antigen on MHC1 as a warning sign, but infected cells will also recognize that, we're infected and secrete something called interferons. These interferons tell other cells nearby that a virus might be coming to infect them, and they should be ready to pounce on that incoming viral genetic material and cut it up. And so it's really useful for us. But Ebola virus stops the secretion of interferon. So neighboring cells have no warning, which of course is going to allow that virus to replicate unchecked. So typically interferon is a good thing. It's part of our immune response, and it prevents viruses from being able to get into every cell nearby. But Ebola prevents interferons from being secreted from infected cells. So that's going to contribute to the enormous viral load that we see with Ebola. Now, a third approach comes back to this idea of secreting glycoprotein. So when we covered virology and the viral structure, one of the things that I hit on was that Ebola is able to secrete a version of its glycoprotein that goes into the bloodstream and just disseminates, not attached to anything. That glycoprotein, which is the antigen, will still bind antibodies, right? But those antibodies are not binding the real virus. They're binding what is the equivalent of a decoy. So Ebola's ability to secrete these glycoproteins that just go out into our blood and basically bind antibodies and suck them up, and then those antibodies are now associated with secretory glycoprotein and not with the real virus, are the equivalent of sending our antibodies on a wild goose chase after decoys. Remember I said that if Ebola can't secrete glycoprotein, its virulence is greatly reduced. And we think that one of the reasons why that secretory glycoprotein is such a stellar virulence factor is because when you finally do make antibodies, some of your antibodies end up going to the decoy version and not the real virus. And so that doesn't help you, that helps Ebola. Okay. So now that you've wreaked havoc in this human, it is time to get out of this human and into the next one. And your best portal of exit is going to be blood. You will have the highest viral load and the best chance of getting into a new human if they handle an infected person that's bleeding out of their orifices. So it turns out that the major advantage to hemorrhaging isn't to cause death, isn't to cause extensive bleeding, but rather to increase the likelihood that Ebola will transmit from one human to another, simply by providing a body fluid that has hundreds of thousands of viruses on the external surface of the body so that a caregiver or somebody handling that body is likely to touch that blood and put it on their own mucous membrane. When people think about viral hemorrhagic fevers, they tend to think that cause of death is bleeding to death. In fact, that they bleed out and they bleed to death. You do bleed, but it's not usually to death. Rather, bleeding or that hemorrhaging is more about transmission than anything else. So people don't actually bleed to death. Again, the purpose of hemorrhaging is to give the virus a better outcome in terms of transmission. So how does Ebola cause hemorrhaging? Well, to answer that question, we need to look at something called the coagulation cascade. Typically, if you have a blood vessel and there's a breach in the wall somewhere, so there's the breach, you induce something called the coagulation cascade to create a clot. You start by laying down platelets that forms what's called a platelet plug, and then you convert a plasma protein that is made by the liver. That plasma protein is fibrinogen, and you convert it into fibrin, and then fibrin, this like fibrous, is going to reinforce that clot. So this happens all over your body frequently. Anytime you get a little tear in a blood vessel, you're going to induce this. Remember that the liver makes fibrinogen. All right, typically this is a localized process, but coagulation can also be induced by a cytokine storm. So when Ebola numbers start to grow, the cytokine storm starts to trigger coagulation. And instead of happening along a vessel wall, it happens inside the blood vessel as shown here. Now, you're going to make all of these small little clots all over your body. And this contributes to something called DIC, which stands for disseminated intravascular coagulation. Disseminated means spread out, intra means inside, vascular is blood vessel, and coagulation is clot. So spread out inside the blood vessel, clots everywhere. That's what that stands for. It is a hallmark feature of all the hemorrhagic fevers. Different viruses have different mechanisms of inducing it. For Ebola, it's a little bit more about the fibrinogen or the plasma proteins. Okay, we're going to build up this clot. We're going to make a whole bunch of these little clots, and then what happens? Well, the clots are going to go into smaller and smaller blood vessels like arterioles or capillaries until they get stuck. Then what happens? Well, blood that's coming behind doesn't have anywhere to go, and so the pressure builds up, and then finally delicate vessels will burst. And so you have little micro hemorrhaging everywhere. And this can be seen in what's called a petechiae rash, which is shown here. Little broken blood vessels everywhere from the DIC. Okay, so then what? Well, now that you've done this everywhere all over your body, you have consumed all of your available fibrinogen and many of your platelets. But your liver is infected and no longer making replacements. So now what? Now you have all these broken blood vessels, an infected liver that's no longer going to be making fibrinogen, or not in any great numbers, and you've already used up all the existing fibrinogen to make these ridiculous little clots that you didn't need anywhere. But now you've got broken blood vessels you actually need to make clots and you can't. So what happened? You just bleed. And that is where the hemorrhaging comes from. You just bleed. And again, the purpose is transmission. So if death does not come from hemorrhaging, what does death come from? It turns out death results from multiple organ failure due to shock, mostly. So there's a bunch of things that are going to contribute to that, but it includes the fact that you infected your adrenal cortex. For those of you that have been through physiology, you'll know that the adrenal cortex secretes both cortisol and also aldosterone. And aldosterone is really important for keeping your salts and fluids in your blood. Without aldosterone, you lose too much via urine, and so your blood pressure drops to dangerously low levels. The fact that you have diarrhea and vomiting only makes that worse. You have an over-aggressive inflammatory response. We know that histamine as part of that causes vasodilation everywhere. Blood pressure drops to dangerously low levels, and then not all your blood vessels are intact anymore either. And so some of the blood is just kind of leaking out everywhere. So again, it really is the perfect storm, and it's amazing anybody survives, to be frank. So it is a crazy, crazy dangerous virus that has multiple ways of really taking away all of your physiological responses and making it so that you just effectively lose your blood pressure and you can't get oxygen to your organs anymore, and then death is imminent. Okay, so I mentioned before that not everybody is going to have the same prognosis, not everybody dies. So what are some things that we note about people who do succumb versus people who recover? So people who do die tend to have non-coordinated and confused innate immune response. In other words, one of the hallmarks that we see is that we see that their early immune system is discombobulated, and we see that their lymphocyte numbers decrease, especially helper T cells. So again, that delays the acquired immune response, you have a discombobulated cytokine storm going on, all of that's going to lead to higher viral numbers and a longer time to bring out your acquired immune system. Whereas people who survive don't do that. They don't overblow their initial immune response. They are unlikely to progress to a cytokine storm. The spears are more likely to go to the castle than not. And consequently, you end up with lower viruses, a more coordinated response, less bystander apoptosis of helper T cells, lower viral load, a faster time to get your antibodies out. Okay, so these are things that we've noted. There are so many similarities here between this and SARS-2 or coronavirus SARS-2. For instance, one of the things that is starting to show up is that some people are getting strokes from, guess what? Clots, coagulation cascades that are set off by cytokine storms. Also, one of the big question marks is, why is it that some people become critically ill and some people are barely symptomatic? What's going on? And I said this when I first started talking about it and we were still in person, and I'm going to say it again. Your initial immune response is critical. If your initial immune response is coordinated and everybody's on board, the virus doesn't really have a chance to cause great destruction. That's true for Ebola, and it seems to also hold some weight with COVID-19 as well. If your immune system is discombobulated or suppressed, again, one of the things that we see with critical cases or fatal cases of COVID-19 is that early T cell counts are really low. Does that look familiar? Looks familiar. Okay, so now that we've covered that, I want to thank you for watching. I hope that this final lecture allows you to really understand how something that is non-living can wreak so much damage in our bodies and apply that to today's current pandemic.